Dear audience, warmly welcome to the scientific evening organized by University of Nova Gorica. Today I am very proud that I can present to you docent Dr. Urška Vrhovšek, who is my dear friend and co-worker for the last times. Maybe a few words about her that are not actually written. She was, this was written, she was born in Celje, but she was there 6 months, then she moved to more Suomi area, to our capital, to Ljubljana, and she spent a lot of her childhood swimming, so Olympia was her best friend, I guess. Afterwards, she decided she will turn to her new occupation, and she affiliated into Biotechnical Faculty of Ljubljana, where she finished her university studies. And then this was not enough. This was not enough, as it is never enough. She went to do her PhD in Universität für Bodenkultur in Vienna, where she was living, if I remember right, in a Slovenian home, just next to the Marienplatz. And I guess she knows Zachertorte quite well from this. For a while, then she was affiliated to the Agricultural Institute of Ljubljana, where she was a researcher after her PhD. But as a lot of researchers, she decided she will try and see how in other places people are doing research, especially agronomical chemistry research. And she went to the Istituto Agrario, S.Michele, Fondazione Edmund Mach in Italy, where she's still a senior researcher and also a head of Metabolomike platform. And I kindly invite Urška to give her talk about metabolomics. Actually, the title is From Targeted Analysis to Untargeted Metabolomics and Application into Biological Samples. Please, Urška.
Good evening to everybody. I'm very pleased to be in Slovenia. I really like to come back and to show what we are doing. So today I will present results of the last maybe 4 or 5 years, and to give you one overview on our work. I'm very grateful to University of Nova Gorica for this invitation, which is giving me this opportunity. So thanks really a lot for this kind invitation. And as Kajetan already told, that I will speak about mainly about metabolomics, which is new approach in analytical chemistry analysis. I just want to show you we are very close in reality. We came today with Kajetan from Trentino, so Trentino is northern Italy, and it took us about 3.5 hours to come to Vipava Valley. Also, it was very nice to be here in such a lovely day. Trentino is a very, very tiny province. It has 500,000 of inhabitants. What is very important for us is that they really invest a lot in research. So the 3% of GDP is going to different research structures, private and public. And what is also important that 96% of Trentino is mountains, so they have only a very few, the area where they can cultivate the I mean, the intensively is very small. So IASMO, where we are now working, and also Kaden is now working with us, is the old name is Istituto Agrario S.Michele all'Adige. It was founded in 1874, so it's a very old institution by the Dieta of Innsbruck, with the aim, in a general aim was to improve the local agriculture, and the main aim of this it was to teach the people about agriculture, to form local farmers, and then it was very important also experimental station in order to help to the farmers on sites. This is the photo, let's say, of the monastery, which still exists. And since 2007, we changed the statuto. So we became a private organization. We have a status of non-profit organization with a private legal status, and we are about 600 people operating in different sites in Trentino. The main aim is research, education, and technology transfer. So we are still very much involved in education and technology transfer in order to help the farmers and to educate. S.Michele is one of the most famous school of agriculture for the children, the kids involved are from 14 to 17 years, and right now we have about 900 scholars. So it's giving a really strong education to all the people who are then working in enology, especially, for example, in agriculture, especially enology. Enology is a really famous school. So now I will speak a little bit about the platforms which were established in our institute. The aim of all these platforms are to study from gene to the function, so to study with genetic and to finish with the product, which is characterized in a big, very strongly with metabolites. So, important part at our institute is genomic. S.Michele was involved in the sequencing of 2 genomes, grape and apple, and now they are also, and strawberry, 3. Then I will learn by the end of the lecture. Then transcriptomics is also very important. We are not involved in proteomics because we would start late, and if you start late, you're always behind. So we decided not to be involved in proteomics. And if we need this skill, then we are collaborating, or we are doing outsourcing and such. I mean, we never want to start late. So this is always, I think, not very good strategy in life, because you will never be able to come before the people who start much before you. Then we are also very much interested in fluxomics, enzymatics, and recently in metabolomics. So we start, like I said, with the gene, and we come to the final product. The platform technologies of FEM are listed here. So it's a strong computational biology platform, sequencing and genotyping, functional genomic, germplasm and collection. We have huge collections in S.Michele. Then metabolomic platform, about which I will speak a little bit more today. We have stable isotope analysis, climate models, cheese and remote sensing. The aim of this platform is that the technology is available to all researchers who needs something. So there is no instrument to person. So like each instrument or collections or so need to be available to all researchers at FEM. And like I said, we start from germplasm and we finish then in metabolomic or whatever it is. So it's nice at the institute because we really cover a wide range. We have many competencies. You can start with the plant and finish with, I don't know, sequencing or whatever you want. So what is the aim of today speech is mainly analytic, and I would like to tell something about targeted analysis. Also, we started with targeted analysis, and these are very important analyses, but are also limiting us a lot. So the current knowledge of chemical processes in plants, animals, humans are mainly based on conventional studies. We are profiling some analytes or some classes of analytes. But with this way, the consequence, the majority which is now esteemed of 80 to 90% of plant, human is a bit better, but not too much, remains unknown. So we do not know. It's really amazing that we probably do not know 80 to 90% of the metabolites which are in plants, mainly because of this approach. So that we were able to start to do untargeted, it was also, it's not only that, the machines were really developed strongly in the last year, so therefore we are now able to use more powerful machines, more user-friendly machines, and this gave us a big step forward. I would like to show you a few approaches we were using, and we are also still using some of them, because not all targeted analyses are now, we will not use them anymore. Some are still very important. One of them are for sure for volatile compounds. As you can see, this is one aromogram. Aromograms are usually quite big, complex chromatograms, and we can annotate many compounds, but still it is a targeted approach. Targeted approach is more or less confirmation science, while untargeted is innovative science. This kind of approach was giving us a lot of information, but like I said before, many were hidden because we just focused on something we wanted to focus. So we left behind many things which were maybe more important, more biological, relevant for one experiment, for example. This is a little bit closer. I'm just showing you these chromatograms to see that we were dealing already before with very many compounds, but all these approaches were still very targeted. We were reading the literature and we said, okay, this could be interesting, this could not be interesting, but we did not allow us to watch a little bit more bright. I was showing you some cases of volatile compounds. Now, this is the same with non-volatile compounds. This is the case of resveratrol. Probably many people heard about resveratrol. It was a compound which was very important for health properties of wine, very modish compound, I must say, a bit abused, but okay, it came out. They discovered it at the right moment when French paradox was published. So anyway, this is the still targeted analysis, simple chromatogram for 3-4 compounds, but also with non-volatile compounds, we can have much more busy chromatograms, much more compounds, and this is the one case of elagitenins in rubus. Elagitenins is not now hot topic in polyphenol analysis, so it is very difficult class of compounds, and therefore remained for us because all the simple things were already discovered before. So we are able to annotate 30-40 compounds in one single analytical run. many of compounds are not known, so it's necessary to do structural elucidation of them. But again, I think this is the last sample case I'm showing you about targeted analysis. So metabolomics. Since we were annoyed of this, since that this is not the right way to go, and we have the possibility to get machines, to invest in people, in personnel, in machinery, we decided in 2006 to change the strategy of the lab and to start with untargeted approach. So we decided to convert a conventional lab into state-of-the-art metabolomic facilities, and in 2006 we had 58 full-time equivalent working in the analytical laboratory. We had two options. If metabolomics would be the part of the existing structure, we could take many advantages of already existing structure, which is the quality control was already established, cross-validation, specific analytical experiences. Or we could also go for a second, a little bit more challenging way, and this was to start from the scratch. And we literally started from the scratch. We were in 2 researchers, in 2 technicians to start. So with no machines, with no personal, with money. This is, and with the vision to go in this direction, this is a lot. So we choose the option B, and we think that this was therefore it was working. It was working because you need new person with different mentality, and we will go for option A. Probably would be, I'm not sure where we will be at this moment. I just put one slide about definition, what metabolomics is. Metabolomics is analytical technique which ideally cover all small molecules below 1,500 Dalton in plant, human, animal, microorganisms. It was so that at the beginning that you inject and you see everything. Of course, it was not so. You need to use different analytical techniques, but not too many. We did not want to go back from where we came. So, and all these metabolites are the final product of gene expression. Of course, metabolomics at this moment is an ideal, and we are trying to get closer and closer to this ideal. It's for sure a complementary study, science, to the transcriptomics and proteomics, because we are coming to the metabolites. So there is, expression of the gene needs to result in metabolites. So there is nothing more to go further on. And its place, for sure, therefore, because of these properties, significant role in bridging the phenotype-genotype gap. Linking each gene to its final product, of course, is not so simple, because one gene is giving more metabolites, and one metabolite can be, it is influenced by many genes, but let's see, we can make the thing a little bit simpler. So about how many compounds we are speaking. As I said, metabolome are all organic compounds in specific plants or also human, vitamins, amino acids, antioxidants, hormones, sugars, aromatic compounds. How big it is in microorganisms, for example, Saccharomyces, also we are speaking about 600 compounds. The simplest one, human, 2,500 plus all metabolites from food, drug. And in plants, we are speaking about 200,000 metabolites per species, should be about 5 to 10,000. All these things are only estimation. We speak about something we don't know in reality very good. So how many compounds per grape, per apple, which are two crops closer to us, it should be estimation about, it's about 5,000 to 10,000 metabolites. And if you put all literature together about grape and wine, about grape, you will maybe find 1,000 compounds, but you will not find 1,000 compounds, even reported in literature up to now. And what is also very important, for example, in diagnostic, only 2% of endogenous metabolites are used. This means that many biomarkers are defined very badly. For example, pancreatic cancer. You almost die. There are some biomarkers, but all patient diagnosis site on the basis of these biomarkers die at this moment. So it's necessary to define much, much better this, and also to find metabolites at the beginning, because nobody is only healthy and diseased or ill. There is a long lag time which we are missing in many diseases. And here is a huge, enormous space for metabolomics to help. So the kind of metabolomic platforms, they are different. I'm not going too much in deep because it's a very technical slide, but basically they are NMR, nuclear magnetic resonance based, and mass spectrometry based. Ours is mass spectrometry based. Each one has positive and negative properties. NMR is for sure good because it's not destructive and it's having a very good library, but it's not sensitive. You just see the most important metabolites. Mass spectrometry has different problems, but it's for sure giving much more, you're getting much more new compounds identified. Structural elucidation is much easier, so they are basically these two ways. You will find NMR or GCMS or mass spectrometry based. So metabolomic platform at FEM is, as I said, mass spectrometry driven. We can also divide it as untargeted, completely holistic approach, and targeted metabolomics. We are using different machines for a different approach. The aim of the laboratory is to use the cheapest machine to obtain the aim. So not to go to use the most expensive machine if you can use in obtaining the same thing, cheaper machine. So for untargeted metabolomics, we are using high resolution mass spectrometries coupled to basically, if we are speaking about volatile compounds, to gas chromatography or to liquid chromatography in case of non-volatile compounds. And then it's targeted metabolomics, which some people are saying that should not be used because according to the definition, metabolomics cannot be untargeted because you need to watch everything. But anyway, we are using it, and these are mainly methods for profiling. So you can quantify the metabolites, and we are working with triple quadrupole machine instruments. These are machines which we are now, we have maybe it's missing some, no, more or less they're all. So for untargeted, like I said, we have one cutoff mass spectrometer, and then we have for GC one TOF. Now we already have one GC per GC, one new machine. Orbitrap is the one on which Kajetan is working, the most expensive machine in the lab. And then we have for targeted metabolomics, triple quadrupole. So. What it was also important for us, the new organization of the lab. So we completely changed, like I said, the point of view. There is not instrument person. There's not, I don't like you, I will not do for your analysis, does not exist at all. All machines needs to run 24 hours per day, 7 days per week. This is what we want to achieve. We are close. The people are already fighting a little bit for machines, but and this is how it must be. So we went from one-to-one to all. So all qualified personnel who can work on whichever instrument. Of course, the school, I mean, they need to be trained, and to work in such a machine, it takes months. But we wanted as many people could use a machine for analyzing their own experiments. And this is very important to form a critical mass of highly specialized analysts. As I said, these machines, you need to be trained, training is long, and they should run their own experiment from the beginning up to the end. Since we have many people working on different machines, we need to have a booking system, so all machines are booked. This is also very important for example, for budget, for rendicazione, to see which instrument was running for which project, for which group, and also to see, okay, this instrument is less used, so we can maybe write a project to use this one more, and this one is completely full, so or we buy a new machine, or we cannot anymore charge more work on specific machines. And there are, of course, as always in life, pros and cons. Pros is for sure that we can have less instrumentation of superior technology. When province decided to contribute to this Metabolomika platform, the first thing which they said, we don't want to have any duplication of machine in the province. So it's necessary to do a specialized center, and not to that. I mean, all machines need to run always, and therefore we cannot duplicate machine in another research center, for example. So higher automatization, and of course, all high tech is really a very stimulative environment for the people to come to learn. We have a very big turnover on the people, but nobody who is living is without the job. So they are immediately fine. They are always going for a better position. So. And what is contra? For sure, it's necessary to plan very good experiments at the time on the machine. We need to form, also because we have quite high turnover in the personnel. And of course, if on a machine more people are working, the machines are having much more black time. So they are broken more frequently. But for us, it's more important that the machines are used as much as it's possible than to use it by one person 8 hours per day. This we don't involve at all. So what is the main topics of interest of the platform now are high throughput targeted metabolomic profiling of different matrices. Then they are involved in the studies which require a systematic system biology approach. We want to find and validate new biomarkers. As I said, most are not known. We want to study very closely the interaction between food and the humans. This is also the project on which Kajetan is working, one of the projects. And then we also want to understand the enzymes, we want to understand the pathways, because metabolomics is not about now to elucidate each compound. This is not the end point. The end point is to put these the metabolites together in new pathways, because if we are not knowing 80% of the metabolites, I think that biochemistry will be needed to be completely rewritten at one point. This is our main project. This is now finally finishing. So we wanted to define grape metabolome. And then we needed to optimize the protocols. The quality control standards are very important. Of course, we have a very strong group of bioinformaticians for establishing data mining pipeline, annotation pipeline. Then we have high throughput application for routine analysis mainly on profiling, and we are applying this to a very large number of very characterized genotypes. This is also very nice in S.Michele because we have from grape up to the end. So if we need one cross, if we need whatever, we can go to the breeder and say we need this, and then they will do this for us. So we start with the grape and we finish with consumer science, because also if we are speaking about breeding for a better fruit, at the end people need to eat this, so they need to taste somehow. And this we are then studying through consumer science. Then now only I will speak about a little bit about the protocols for metabolomics and application. Metabolomics is about 50 years behind genomics. So we need to establish, each lab is establishing the protocols. The machines are different. We are dealing with so many different classes of compounds. So basically, we need to start very much from the beginning. And then I will explain, I will show you a few applications, which is what we really want to do. Because metabolomics is a tool, and we need to now, with this powerful tool, go back to the real biological problems. So at the beginning, it was necessary, for example, I'm speaking about the grape, to establish the extraction protocols, because if we want to define the metabolome, we need to extract as much as possible. So it's necessary, already this is an issue, what we will take, so not to leave away completely one class of compounds. And we were working on this about half a year. So now we are finishing up with 3 fractions in order to cover as many metabolites as possible, with lipids in chloroform fraction, and then with polar and apolar compounds. And adding to these 3 also volatile compounds, we are working basically with 4 analytical methods. And we are able to annotate for now about 2,000 compounds. Okay, this is a very technical one. Then, of course, it's necessary to develop also all separation methods. As I said, in the contrast to genomic or proteomic, we are dealing with very different classes of compounds and very many compounds also. So we established basically 2 methods. One is a short method, it's a profiling method in order to do many analyses in a short time. And then we have a long method, which is longer, of course it is long, because especially for HPLC, UPLC, here we still have an issue because the separation power is not so high. So one analytical peak, such a peak, can be 10 compounds easily. So it's not like gas chromatography when one compound one peak is 1 compound. So, and then you need to figure out what is here, everything below. So therefore it's necessary, we need also longer methods. So the same is also with gas chromatography. We change a little bit now, we are changing now the protocols because we were not very happy with this machine, GC-TOF, and we are now sacrificing high resolution for two-dimensional chromatography. This is now the fashion. I think also the people are more and more working in two-dimensional chromatography. And what is very annoying work, and you need to have a lot of patience to stimulate day by day the people, is to inject all commercially available standards. This is not giving any publication. It's just to construct the own libraries. So this took us about 2 years of work and about 100,000 euro only for standards. But since there are so many unknowns, I mean, you need to get rid at least about these known compounds, because otherwise you don't know where to start. If you need to structurally sedate also the known compounds commercially available, then you do a lot of work which is not necessary to be done. Anyway, we are now trying to, we'll see, sell, I don't know. We don't want to, that everybody needs to do this job, especially for, we were never understanding why the companies are not doing this job, at least for primary metabolites, because they're everywhere. So, and it's like something like NIST in GC. And I mean, this is really a job which should be probably, it will be at a certain point done also, like they will sell machine with application with the commercial library. This is, I think, very important, because we were able to do this job, but it could be overcome much more easier if somebody, let's Waters or Thermo, whoever, would sell already this together with machine. For now, yeah, we needed to do this. There are also some open source library like human metabolome database, Saccharomyces database, metabolome database maybe, but this is only a help. So you need always to be 100% to annotate correctly, to be 100% sure that what you're doing is correct. You need to do your own libraries. And this, yeah, we did this for all instruments, all machines, everything. This is again one application, for example, this is in GC, you have different chemical analytical parameters which you need to put together. So there is a lot of compromise also, because of course you cannot analyze everything. It's crazy, you cannot. But you need to find the way to analyze as much as you can. So again, these are different. I am showing this slide only to show you that there are very many different analytical parameters which you need to taste. This is the method of targeted metabolomics for primary metabolites. For targeted, we are doing, for example, short runs, as I said, about 140 compounds in one analytical method, 15 minutes. And the pro of targeted is that you get quantitative results, and you can compare year by year or two different experiments. While with untargeted, it's semi-quantitative. You can compare, I don't know, the group of diseased and healthy person, and you see the biomarkers which are different, which is up and down regulated, but you cannot give one number like this is 5 and this is 3. You can say this is more and this is less, for example. We have all these methods on both, on targeted and untargeted machines. This is now the most in method in our lab. Also, some people from your university are just coming to do some experiment on it. So, like polyphenols are very important, you will be healthy and so. And we were developing one method which we are really happy with, which is covering a wide range. We are again about 150 phenols, and characterizing different fruits, or also we are doing something also on metabolites and catabolites of polyphenols after eating the fruits. And what is important is that we are showing these methods are constructed in a kind of models. So if you are analyzing, I don't know, raspberry, you put the box with compounds which are in all fruits more or less, and then you're adding the box where you can, of the compounds you know that they are present in raspberry. But we are also going a kind of untargeted, because these are very sensitive machines. We found many polyphenols never reported in some fruit, for example, resveratrol in apple. Resveratrol was never reported in apple, and or fluorescein in grape or something like this. So also here, despite going targeted, the power of machine is so big that we can also analyze compounds never reported in certain matrix, and this maybe is only academic research. Maybe this does not have any really nutritional value, but it's interesting also for academic research. So this is the same method. As I told you, it is modulated by modules, and we are expanding it, of course. If somebody is coming with Arabidopsis, I'm the first thing telling you, tell me which compounds you would like to add, because this, so then it's important to, it is kind of flexible, so you add, you take, you analyze what you want, what is important for you. So, and it's working very good. Like I said, we were adding also some metabolites, catabolites of polyphenol metabolites and catabolites which are circulating in bloodstream, and I must say that it's really very, very sensitive instrument, and we are getting very interesting results. For now, I have one PhD student who is working on blood-brain barrier. I will show a little bit later, and he's really getting very interesting results. The method which is also important are lipids, especially we are now starting to study resistance. Lipids are, according to me, very important in plant resistance. It's one of the most important, almost unknown. And why? Because when we are doing human perturbation studies, whatever, or mammals, the first class of compounds which is reacting is always lipid. And I'm almost sure that it's the same also in plants, because why the plant should react completely different than human? No, it's not. This is one study we were doing with Andrea Vanza from Agricultural Institute. It's one untargeted approach. I would just like to show you, how many different compounds you have, nothing more, because, and then usually in untargeted, like I told you before, you get two classes. So the most distant biomarkers in two groups are the most distant. But it's not said that they're the most biologically relevant. This is another problem in metabolomics. You see that they are changing a lot. And then if you are really sure that this is the biomarker, you usually need to do isolation, NMR, and then to do some mechanistic studies, cell culture, or something like this. Because the problem is that this is only a hint. But then you really need to demonstrate that this compound is really biologically relevant. This is the study I was mentioning just before. We are studying blood-brain barrier. For example, there were many studies that small berries are important were showing very good results in behavior studies of rats, for example, adding the small berries to the meals. The rats were behaving better. The one which were developing Alzheimer were developing slower, or were even sometimes recovering, despite being a progressive disease. But nobody was really showing that these compounds are passing blood-brain barrier. So you need to prove that these compounds are coming really to the brain. And what is important is that you are not doing this analysis with the compounds which are present in berries, because these are not the ones which are circulating in blood, but in blood are circulating metabolites and catabolites of these compounds. So this is was necessary then to isolate or to synthesize. Some are commercially available. So we are studying this with both targeted and untargeted approach, always in parallel. Also because targeted is also much more sensitive, untargeted is more innovative, but for ethical reasons. So if we are sacrificing the animals, we are trying in one data set to get as much data as possible. And here is written more or less. The strongest point is accurate quantification in targeted and in untargeted are new biomarkers. This is the study we are doing with Melita. Like I said, for somebody, metabolomics is a science. For me, metabolomics is a tool. And I want to go back to everything that we were doing before, with much more powerful tool. So with Melita we are studying canopy microclimate manipulation in Pinot Noir, and we are, probably as good as me what she's doing. And so therefore I will not go in detail, but we are also tracking this now with targeted and untargeted approach. And it's a very nice project, and our institute is very interested, therefore we are doing in parallel in S.Michele in here, because it's exactly where we want to go back to the vineyard, to go back to whatever, and to do, to study the same biological problems with new approaches. So this is something what we are doing with Jure. Here Sofia is also involved. We are studying genes behind aroma compounds in wine. Again, targeted, untargeted metabolomics. This is more on volatile compounds. And non-Saccharomyces yeast also. I have another PhD student who is also working in non-Saccharomyces yeast and on wine, and I think this is also where we can come very close together, because we have we don't have completely different, the same, but we have very similar interests. And I think this is going to be a very nice, promising project. We are putting together the competencies of two labs, which are fitting really very good together. Then we have also imaging technique. Sometimes it's very important to show the localization of this is one polyphenol, where exactly is localized, and so this is targeted approach, because we need to know what we are looking. But for example, there were some metabolites which were always reported to be in skin of, for example, in skin of apple, and we saw that it's just below the skin, because this is always many polyphenols in apples are in skins, but many people are then peeling the apple, so they are not anymore eating the polyphenols. So imaging is a very good technique to show the localization. It's very good technique also to study resistance, because usually the response of the plant is necrosis, or something is happening on this place. So you can show where exactly which compounds and where, because once you're doing the extracts, you lose completely the information about the localization of these compounds, but it's many times very important. And I'm finishing, so to see a little bit more globally what we are doing. So we are basically going from omics to improve the fruit quality, so like assisted breeding. So this is our main aim in agricultural science. So we are going from classical breeding to modern breeding, developing and validating tools for breeding programs, and we need different steps: metabolite profiling, fluxomics to understand what's going on, functional genomics, and then of course on this side is important fruit quality, nutrition, antioxidant capability, bioavailability, and sensory. Sensory is very important, because you can develop one very nice apple, but if nobody is not eating it, then it not did too much for the assisted breeding. So, the main issues are nutritional value, shelf life, resistant, and pigmentation. And this is another very nice project. We have very strong nutritional nutrigenomic groups, and they are studying basically typical fruits and cheese from Trentino. Small berries. Trentino is the biggest producer of small berry in Italy, also because I said 96% of the area is mountain, so we cannot have like Australia style wine production. And we are constructing now, we have a very good model of gut. We are integrating metabolomics and metagenomics, and performing also dietary intervention studies. And because the aim now is not anymore to live 100 years, is to live 80 years, but live or whatever, but to come almost to the end healthy. So this is the most important point, because it's useless if you're last 15 years in the bed. So, and this is also what this project is about. And I stole this slide from Melita, but I really like it. I've never found such a good phrase. And this is of course true, that to understand the whole, you must study the whole. If you just focus something, if you just do a small slide, you will lose a big story probably. And the goal of system biology and metabolomics is just to do that. And with this last slide, I would like to thank you for your attention.
I would like to thank Urška for this very nice presentation and brief insight into metabolomic methodology. If somebody from the audience have any questions, please let me know. No, then maybe a few questions, brief questions from me, from my side. First, do you think, or let's say, let's be a little bit more optimistic, when do you think we will be able to reach 90% of metabolites in, let's say, grape, apples?
I think that now it will start to go more quick, especially if the people will put the data together. So, I mean, I don't want to do predictions, but I think that 10 years, maybe. We'll go, I think we'll start to go, maybe a little bit more, because necessarily, but what is the most time consuming is to do isolation of compounds, because you need to, at the end, we are also doing you're isolating many compounds, because the problem is if you have, like isomers, cis trans, you can have master solution on million, but I mean you will never be able to, and they can be biologically completely different. So, but yeah, I mean that I think that we'll go now relatively fast. Yeah.
Do you think one of the main obstacles in genomics or genomic analysis, it was the actually data processing, and now when I'm dealing with this, I also see many weak points in the speed or accuracy of the processing data, so this field will be most probably also have to be developed a little bit more.
Yeah, of course. The data processing is for now, for sure, a bottleneck, because everybody, I mean, each institute, each group is starting basically from zero. So we are not using too much commercial software, so I think that will be a long way to go. Yeah, but I think that the longest is for sure structural sedation of compounds.
And then I would have another question, which is a little bit maybe practically oriented. I noticed or that one of the main topics of the research in the next few years will be based on the resistant varieties, which is also being quite a lot effort was put in the University of Udine, and also here. And I am interested about your opinion. Do you actually think that, let's say in 10 years, the resistant varieties will change our varieties which are planted now in, I don't know, Trentino, Vipava Valley or whatever?
No, will not change. I mean, it is a really important topic, because if you need to spray the apple 25 times, or the grape, whatever, or if they're spraying, we know that they're spraying small berries almost up to the when they are going to the market, and then you're claiming that they are very healthy because they're containing lagidenins, is not very ethical sometimes. But for sure it's very, very important way to go because of also ethical, but especially economical reasons, and because I mean this is really a cost. But I think will be a long way to go. In Italy they are already planting resistant varieties, and the quality increased enormously in the last 5 years. I mean, I'm doing the tasting of this because it's really my personal interest, and because at the beginning they were resistant, but the wine was crap. So the same problem, like if the apple is healthy, but nobody's not eating it. But now, I mean, some Cabernet Cortis, there are really some varieties which are really good. So yeah, I think we'll go. Only that I don't think it will be very easy to convince winemakers. I mean, it will be some part of wine production, or also apple production for sure. Yeah.
But I think this could be also one way to go also in Slovenia and Vipava Valley.
You need to convince. Yeah, I don't know how is the legislation, because sometimes the people even want to plant, but it's forbidden. So it's necessary. Here is very much about legislation, how much you can influence their changing, they're allowing these things. So in Trentino now, I think they are allowed to plant 2 or 3 varieties, and that's all. Despite there are some very promising. There is Solaris, Brunner, of red, maybe there is not even one at the moment. But yeah, I think that this will be very, very important topic. I mean, our institute is having like this in Italian progetto bandiera, and we are really working very, very strongly. But especially to come with new breed, it take you 10, 15 years. If you start today with the like crosses, you will maybe finish in 15 years on the market with one apple.
So when we will be retired.
For sure.
Thank you once again for
your
lecture, and let's go to some sensory metabolomics.
Not resistant varieties for now. Next time.
Thank you.
Thanks to you.